research focuses on ovarian cancer, which is still the most lethal gynecologic malignancy in women. And even though we call it ovarian cancer, my take home message for today is that's probably really bad branding because it is now recognized that the most common and deadly type of ovarian cancer, called high grade serous, really arises in the fallopian tube. Now, why do we care about this? Well, first of all, imagine you're a patient at high risk for developing ovarian cancer because you've had your genome sequenced and you recognize that you have BRCA1 or 2 mutations. Instead of removing the ovary and inducing infertility and surgical menopause and leaving the cells behind that would give rise to cancer, we can now do a surgery that removes the fallopian tube instead, preserving options for IVF, retaining circulating hormones, and actually removing the cells that give rise to this cancer. When we're doing drug discovery in the lab and we try to look at enzymes or receptors that are overexpressed or tumor suppressors that are lost, and we compare the tumor to the wrong epithelium or the wrong normal progenitor cell, we might completely misunderstand what's a good drug target or what's something that we want to re-express in order to mitigate this disease. And if we're trying to do early detection, which by the way, there's still no early detection for ovarian cancer, and you shine a bright light, develop a magnetic resonance imaging tool or a FRET probe or a new PET imaging scan, and you scan the ovary, you've completely missed where the early disease is forming. So this is the reason it's very important to understand this. Now, what can we do about it? There are a few risk factors associated with ovarian cancer, and one of the most important ones is ovulation. So things that block ovulation, like birth control pills, multiple pregnancies, lactation, early menopause, are all protective. But studying ovulation is very difficult. As you can imagine, the ovary is right next to the fallopian tube and it's contributing or disseminating these little signals that are driving the cancer process in the tube. But how do you capture those, or how do you study their concentration gradients in that tiny space? So our lab has developed microfluidic perfusible organ-on-chip technology where we can actually ovulate ovaries from mice on the platform and study all of the secreted factors in real time as they come off and bathe the fallopian tube, or models that we generate in the lab made from fallopian tube cells that represent early disease. And proximity matters. So it's not the entire tube, it's just those tiny finger-like projections that touch the ovary where these early cancers form. And they love the ovary, which is why we call it ovarian cancer. It's a fertile soil that allows for the spread of these cells into that tissue where they ultimately disseminate and seed the entire peritoneal cavity. So we can use a technology called imaging mass spectrometry to image the chemical concentrations between models we make of ovaries and fallopian tubes. And we've identified a story where a spark, not a UIC talk, but a serine protease, remodels the ovary to secrete hormones, norepinephrine in this case, driving beta-adrenergic signaling and metastasis, and link it to data showing that epidemiologically, taking beta blockers protects against ovarian cancer spread.